Assalamu alaikum. Welcome to my YouTube channel. Today the topic of my discussion is glomerular filtration. Glomerulus is actually a network of capillaries and it is the very first part of nephron. In the structure of nephron, after glomerulus there comes Bowman's capsule, then proximal convoluted tubule, then loop of Henle, and then distal convoluted tubules, and finally the collecting ducts. So the major function of glomerulus is in filtration of blood. It releases its filtrate in Bowman's capsule. The filtrate that comes in Bowman's capsule after being filtered by the glomerulus is identical to the blood plasma in its electrolyte and water composition. It means that the medium to high molecular weight proteins and other components are not allowed by the glomerulus to be filtered and to come in the glomerular filtrate which is being released in the Bowman's capsule. As I have told you that the function of this glomerulus is to filter blood, so it is very important to know the pathway of the blood that comes into the glomerulus. The renal artery that enters the kidney branches into smaller arteries called afferent arterioles. These afferent arterioles then divide into glomerular capillaries, and then these glomerular capillaries are drained into efferent arteriole. So in this way, on both sides of glomerular capillaries, there are arteries, and we know that the blood pressure in arteries is very high, and this higher pressure is responsible for the filtration of blood in the glomerulus. This hydrostatic pressure in glomerular capillaries is very high, and it is about 60 millimeter of mercury, and this causes the filtration of blood in the glomerulus. Glomerular capillaries have three layers, and these are endothelium, basement membrane, and visceral epithelium. Endothelium, as we know, is the innermost layer of any type of blood vessel, and in case of glomerulus, it is a single layer of cells and it contains thousands of holes, and these holes are known as fenestrae. And the size of these fenestrae are 70 to 100 nanometer. So they act as sieve and provide channels for the passage of water and non-cellular components from the blood to be filtered and to go to the basement membrane of glomerular capillaries. Next to this endothelium comes the basement membrane, and this basement membrane is made up of most importantly type 4 and type 5 collagen proteins, proteoglycans, laminin, fibronectin, and intectin. Basement membrane further contains three layers, and these three layers are lamina densa, lamina rara interna, and lamina rara externa. Lamina rara interna and lamina rara externa actually sandwich the lamina densa. It means that lamina densa is sandwiched between lamina rara interna and lamina rara externa. Lamina densa is a thicker layer or dense layer, while lamina rara interna and lamina rara externa are thin layers. Lamina rara interna is present on the endothelial side of the glomerular basement membrane, while lamina rara externa is present on the epithelial side of glomerular basement membrane. So, if we talk about the layers of basement membrane in sequence, then first comes lamina rara interna, then lamina densa, then lamina rara externa. After endothelium and basement membrane, there comes the visceral epithelium. This visceral epithelium is composed of interlocking cells known as podocytes. These podocytes contain food processes, and between these interdigitating food processes of podocytes, slits are present. And these slits are known as filtration slits, and their size is 20 to 30 nanometer. So the fenestrations in the endothelium and these filtration slits in the visceral epithelium function as channels for the filtration of blood in the glomerulus. So we have studied that glomerular capillary wall is made up of three layers: endothelium, basement membrane, and then visceral epithelium. This glomerular capillary wall acts as a barrier to the forces favoring and opposing the filtration of blood. Forces which are favoring the filtration are hydrostatic pressure within the capillary and oncotic pressure in the Bowman's capsule, while forces that are opposing filtration are plasma oncotic pressure within the glomerular capillary and hydrostatic pressure in the Bowman's space. Hydrostatic pressure is actually the pressure exerted by the blood on the walls of capillaries. Oncotic pressure is the force that is exerted by the proteins that are found in the blood. So this hydrostatic pressure will act to filter the blood, and the blood will push against the walls of capillaries so that it can be filtered through the glomerulus. The other force that favors the filtration is the oncotic pressure in the Bowman's capsule. And we have seen that medium to high molecular weight proteins and other blood components are not filtered by the glomerulus. And as the oncotic pressure in the Bowman's space is the force that favors the filtration, and the high molecular weight proteins are not filtered, so this oncotic pressure is not much effective in favoring the filtration process. On the other hand, oncotic pressure is much effective in the glomerular capillary, and therefore it acts as the opposing force for filtration in glomerular capillary. Now come to GRF. It is the milliliters of glomerular filtrate formed per minute per kg of the body weight. And this glomerular filtration rate depends upon the permeability of the filtration barrier and surface area available for the filtration. So the permeability of the capillary wall or this filtration barrier is determined by the chemical and structural characteristics of the capillary wall or this filtration barrier. These structural and chemical characteristics of the capillary wall make it a selectively permeable membrane. And due to this selective permeability, this capillary wall is responsible for the differences in the rate of filtration of the blood components. As I have mentioned before, that the glomerulus doesn't allow the blood proteins, and these proteins are not present in the glomerular filtrate. The reason for this is the size of fenestrations, that is only 70 to 100 nanometer, while the size of blood proteins is in micrometers. So these fenestrations don't allow the blood proteins to be filtered from them. One more important thing to be mentioned here is that the size of albumin protein, which is present in blood, has size nearly similar, so that it can be filtered through the glomerulus. But in normal animals, the albumin is not filtered and it doesn't come in the glomerular filtrate. So now the question is, why this albumin is not filtered by the glomerulus? Its reason is the charge selective barrier in the capillary wall. Let's know what is this charge selective barrier. There is electronegativity on basement membrane, endothelial cells and visceral epithelial cells. So in this way, whole filtration membrane is electronegative in nature. And we know that albumin molecule is also electronegative, so it is repelled by the charge selective barrier of the capillary wall, and therefore albumin is not filtered by the glomerulus. So the structural and chemical composition of the capillary wall is such that the cationic forms of many substances are freely filtered than neutral forms. And these neutral forms of the substances are freely filtered than anionic form of the same molecule. In short, this charge selective barrier filters the cationic forms of many substances easily. Similarly, small sized molecules are easily filtered by these capillaries. The shape and deformability of the molecule also affects its ability to cross the filtration barrier. Deformed or irregular shaped molecule will find difficulty to cross this filtration barrier. So finally we reach the conclusion that blood cells and plasma proteins are not filtered by the glomerular capillaries because of their large sizes. And that's why the glomerular filtrate is identical to the blood plasma in its electrolyte and water composition.